Welcome to Arun Biology Forum. So today we are going to see about simple staining process. So the simple staining is used to color the bacteria, and it helps to view under the bacterial cell shape and structure and its cellular arrangement under the field of microscope. So first we will see what are the materials required for the simple staining. So you need the cell culture, you need the bacterial plate, then you need cotton, then slide, then micropipette, then water for dilution, and the staining solution. So now let's start. First we have to clean the slide. So using the 70% isopropyl alcohol. First we have to clean the slide. So clean the slide using the isopropyl alcohol thoroughly. And we have to allow it to dry. Just let's place a slide over here. So using the inoculation loop, first sterilize the loop. Going to see about the simple staining of bacteria. So for that, first you have to take a glass slide, and you have to clean that glass slide using the 70% isopropanol, and leave it for air dry. And then what you have to do means you have to take small quantity of water, aseptically, and you have to place the water at the center of the slide. Okay, center of the slide. So and after that, so what you have to do means you have to heat the inoculation loop. And after heating the inoculation loop, pull the loop for 20 seconds, and then you have to take a loop full of bacteria. So you have to wait for 20 seconds for allowing the loop to get cooled down. So now it's cooled down. So aseptically take some quantity of bacteria, loop full of bacteria, like this, and just mix it with the water. the bacteria with the water. Mix it thoroughly. So we have to thoroughly mix it so that the cells can be distributed uniformly over the along the smear. Okay, so now it's spread evenly. So now heat the inoculation loop. Heat the inoculation loop thoroughly and keep it aside. And now the important step is we have to dry the prepared smear. So we can dry this smear by passing slightly over the flame repeatedly like this. So this is the process of fixation of cells. So this is a procedure of fixing the cells. So after fixing, just keep the slide aside and let it cool for a few seconds, and after that we will add stain. So now we can switch off the flame and we can keep it aside. So this simple staining procedure is mainly used to stain the bacteria to know the shape, mainly whether it is a rod shape or cocky shape, and to find the arrangement of cells, whether it is staphylococci or streptococci like that, or diplococci like that. So if it is a rod, means shape, regarding the shape we can, this is a very simple experiment. Using this experiment only we can know about the shape and arrangement of cells. We cannot predict whether it is a gram negative or gram positive. For that we need another staining process called gram staining. Okay, so now we can add the staining solution. So now we can add the, using the pipette itself, we can add the staining solution. So you have to add the staining solution. You have to add the staining solution. The staining solution should spread all over the, or simply you can pour like this. It should spread all over that area. It should spread all over the smear like this. So now it's spreaded all over the smear. Okay. So now allow the stain to stand for 20 to 30 seconds, and then we will wash it, and then we can view under the microscope. Okay, so this is a simple staining process. Okay, so now the staining time is over. So now we have to clean the, not clean, wash the slide with water. We have to wash the slide in water. thoroughly in. So you can also use either you can use the tap directly, or you can use like this also. Now the washing step is over. So now we have to allow the slide to dry completely. Place it here and allow the slide to dry completely. So the final step is, after drying the slide, we have to observe the slide under the microscope. So microscope, we all know this is a compound microscope. So down there is a mirror. From the light source, the mirror reflects the light and it sends the light through the condenser lens, and it reaches the objective. Before reaching the objective, it crosses the sample and the objective lens, and we can get the magnified image. When we observe through the eyepiece, we can get the magnified image. Okay, so this is all about. So this is the focused bacterial image. So here we can see the rod shaped bacteria. So it is stain as violet color because we used this as stain crystal violet.